Dobro večer in lepo pozdravljeni na še enem v seriji od znanstvenih večerov Univerze v Novi Gorici. It's my great pleasure today to introduce our speaker today, Dr. Lawrence Banks from International Center for Genetic Engineering and Biotechnology. Dr. Banks is head of the tumor virology group, which is one of the leading group in the human papillomavirus research, we can say. He's also a great supporter of our molecular biology research at the University of Nova Gorica from the beginning, from the establishing of this small group still in the frame of laboratory for environmental research and all the way till now. Dr. Banks graduated from Leeds University in UK in 1984 with a PhD thesis on herpes simplex virus proteins and viral replication. He moved then to the Imperial Cancer Research Fund in London, working with Lionel Crawford on P53 and human papillomaviruses. In 1988, he became an independent research scientist at the Ludwig Institute for Cancer Research in London, where he continued his studies on the role of human papillomavirus oncoproteins in the development of cervical cancer. Due to his very successful research work, he was very early appointed as a group leader of the tumor virology laboratory at ICGEB in 1990. During this time, or since then, his laboratory has become internationally known for its work in human papillomaviruses and their roles in the development of human malignancy, and more recently on the HPV infection and in relation to host intercellular trafficking. His group published over 150 papers in peer-reviewed journals, and more than 10 students have graduated from the laboratory. Dr. Banks is also one of the main organizers of the annual DNA tumor virus meetings, and the one this year is being organized in Trieste for the 4th time already. So with this, I would like to finish my introduction, and I'll give the word to our guests and the lecture about what is current knowledge about human papillomaviruses and their oncogenicity.
Okay, thank you very much, Martina. I'd just like to take this opportunity to thank particularly Danilo for the very kind invitation to come to give this presentation in what is really a fantastic location. It's a very beautiful place and it's a glorious part of the world, so thank you very much for giving me this opportunity. Before I start, I'd just like to say a little bit about the organization that I represent, the ICGEB. It's an international organization. We have 3 research centers located in Trieste, New Delhi, and Cape Town in South Africa. And there are over 60 countries that are members of the ICGEB, and of course, Slovenia is one of those. And what we do is a variety of things in terms of really just helping scientific development. We offer a variety of long and short-term fellowships, and as part of this, we have a very active and successful PhD program. We also organize a variety of meetings and courses and workshops, and these can be held both within Trieste, in our component laboratories, but also in our member countries. So if anybody here is at all interested in organizing meetings at any point, you should consider contacting us for potential support. We also have a research grant program where we fund research in our different member countries. This just shows you the current distribution of grants worldwide. So again, if any of you are interested in any of these programs, please feel free to contact me either at this email address, and you can also get more information on our website. So, back to the topic of tonight's talk. Viruses and cancer. Approximately 20% of all human cancers are a result of infection, and 12% are actually due to viruses. The viruses that cause cancer in humans are very diverse. They span the whole evolutionary spectrum. And they've been with us for quite a long time, with Epstein-Barr virus being the first tumor virus to be described. And the last one to be identified, the most recent, is Merkel cell polyomavirus, which causes a rare form of skin cancer. But what I'm going to be talking about today are human papillomaviruses. And why are human papillomaviruses so important? Well, they actually are responsible for 5% of all human cancers. So this is actually a really very significant number. Cervical cancer is by far and away the most important, and these viruses are responsible for approximately 100% of these cases. They also cause a significant number of other anogenital cancers, and they're also responsible for the development of head and neck cancers at defined anatomical sites, and this proportion is increasing rapidly. So globally, these viruses cause around 600,000 new cancer cases each year, and there's a group of different papillomaviruses, approximately 12, that all of which have been defined as cancer-causing in humans, and the most important type being HPV-16. The distribution of cervical cancer is global, but you can see from this distribution, which is shown overlaid on the map showing the ICGEB member countries, it's a particularly devastating problem within developing parts of the world, and particularly in Southeast Asia, where the incidence rates have reached epidemic proportions. Now, HPV-16 accounts for around 60% of these cervical cancers. It's a relatively small virus. It has a double-stranded DNA genome, and really it can be just divided simply into 3 regions. There's early gene products that are expressed early in the viral life cycle. There's 2 late gene products which comprise the virus capsid. This is the structure that encases the viral DNA. And there's a non-coding region which regulates the expression of these different viral gene products. Now, the virus, its replication, its life cycle, is intimately linked to the differentiation of the infected epithelium. So in the skin is formed by, initially you have these so-called basal cells, and these are the only cells in the skin that can divide. These divide. Once they divide, you give a daughter cell, and these daughter cells begin a process of differentiation and move up to form this epithelial layer. Now, all papillomaviruses require this differentiation process to be able to replicate. Now, the virus is believed to enter the skin through microtraumas, small lesions in the skin. It gains access to these basal cells. And then as this differentiation occurs, you see coordinate expression of the different viral gene products. And ultimately, this gives rise to new infectious virus particles, which can then go on and infect other people. Now, the major point about this life cycle of these viruses is that the papillomavirus does not have enzymes that can replicate its own DNA. So if it wants to replicate its genome, it has to use the cellular machinery. Now, all of this region of the epithelium where the virus replicates the DNA, these cells have normally exited the cell cycle. So there's no enzymes present which would normally replicate DNA. And this is where the 2 viral oncogenes, E6 and E7, come into play. They create an environment here that is favorable for DNA replication. So they push the cells into a replicative type phase. And so what normally happens over a period of 2 or 3 weeks, you get virus DNA amplification, you produce late gene products, and you ultimately produce new virus particles. For reasons that are still unclear, you can have individuals that can show persistent infection. So that means people can be infected with this virus, and you can see evidence of continued virus replication, virus production occurring for several years. Those individuals, in those particular situations, this is the biggest single risk factor for ultimately going on to develop a malignancy. This can take place over many years, and there's a series of additional events that are required, before malignant progression occurs. But the standout feature of the development of these tumors is the continued high-level expression of these two viral oncoproteins, E6 and E7, and these drive this malignant progression and ultimately resulting in an invasive carcinoma. The important thing to remember about this process, this is a complete mistake as far as the virus is concerned. What the virus wants to do is this: produce more virus particles. When this occurs, there's been a breakdown in this replication process, and you get the events which result in malignancy. And to emphasize, these lesions are not infectious. There's no infectious virus particles present. So this is what these lesions look like. This is a cervix, and you can see here this very small, flat lesion. It's really barely detectable to say that this is a problematic lesion. If you look at this and you stain the epithelium, and you stain in red for cells that are replicative. If you look here in the uninfected part of the epithelium, you can see the only cells that are dividing are really along this basal level. So it's just here where cells would normally divide. When you have the papillomavirus present, you see expansion of these replicative cells. So you're increasing proliferation within the epithelium by this papillomavirus. At the top here, shown in green, are late viral gene products. So these are cells that are producing new virus. These are infections. And so this will be a normal infection with HPV-16, and it will be what we will call a low-grade lesion. It was not particularly dangerous. It's just a normal viral life cycle. However, as you get this progression to malignancy, you can look here. It's the same cervix, but there's another somewhat more prominent lesion. And this is actually a high-grade lesion, a lesion that is beginning to progress towards cancer. And when you look at this lesion, it's quite striking. You see many, many more cells that are proliferative, and you see virtually no cells that are actually completing the viral life cycle. So this is a very nice example of what happens when you go from a normal virus infection through to processes which are ultimately going to progress and develop into malignancy. This lesion would need quite urgent intervention to treat this, to prevent the development of a fully blown tumor. So this is just really a model of how we think these papillomaviruses infect and cause cancer. You get infection through microtraumas to get to these basal cells. In a normal infection, you see the viruses maintain as small circular episomes. These get amplified, and you produce new virus particles. And this will be a classical low-grade lesion, SIN1 or SIN2. These infections normally get resolved. It can result in clearance, but more often it's likely to result in latency. And this is something which is still an active area of research. In cases where you get persistent infection, you have this whole process going on for many, many years, and you can get initial changes take place within cells, mutations within the host genomes that can ultimately result in an expansion of these cells and produce a high-grade lesion, ultimately resulting in the development of a malignancy. So we know that the two viral oncoproteins, E6 and E7, drive this progression to the development of a tumor. And we also know from a whole variety of studies that those cells that are transformed, that are cancer cells, have to have these two proteins present. So you can take something like HeLa cells, which I hope some of you have heard of. These are cells that were taken from a cervical cancer in the 1950s. They grow perfectly in the laboratory and have continued to grow for 50 years. So you can imagine all the changes that have taken place in those cells. If you block the expression of the E6 or E7 proteins in those cells, the cells will stop growing. So that's pretty amazing. It tells you that even after all that period of time, all the abuse that these cells have had in tissue culture, these two viral gene products are essential for maintaining that proliferative potential. So they do represent excellent targets for therapeutic intervention in HPV-induced malignancy. Now, in terms of, I'm sure all of you have heard of the vaccine that's available to papillomaviruses. There are two currently on the market. One is a bivalent, which covers HPV-16 and HPV-18. Other is a quadrivalent, which also covers two benign viruses, HPV-6 and 11. So why do we still need therapies if there's a very good vaccine? Well, if we globally vaccinated everybody today, it would still take 10 to 20 years before we impacted upon the disease. There's major issues in terms of cost and logistics, particularly in many of the countries that really need the vaccine. And the vaccine is not therapeutic. It's only prophylactic. So many millions of women who are currently infected with the virus will still go on to develop cervical cancer. And the WHO estimates that the numbers are actually, in fact, despite the vaccine, likely to increase quite significantly over the next 10 years or so. And it's also important to remember that there are many different papillomavirus types that cause cancer, but only a very small subset of them are actually covered in the current vaccines. So with that sort of general sort of background to the disease and why we're interested in this virus, what I'd like to do is tell you two stories. One relates to our interest in how does the virus actually get into the cell in the first place. And then the second story that I'll tell you about is how does the virus drive this development of the tumor. So when you consider entry, the first thing you have to think about is the virus itself, the virus capsid. As I mentioned at the beginning, it's only comprised of two viral proteins, L1 and L2. L1 is the major component of the capsid. And actually, this is a fantastic protein. You can express L1 just by itself, and it will self-assemble to make the virus. So it actually looks indistinguishable from a native papillomavirus. And it's this L1 protein which is actually the basis of the vaccine. So you just express L1 by itself. It forms these so-called pseudovirions, which you then can use for vaccination. So L1 is the major carrier of all the genetic information. But the one that we've been particularly interested in is the L2 protein, which is a minor component of the capsid. And it's the key player, however, that determines that the viral DNA can make it into the nucleus of the cell. So once the virus gets into the cell, L2 becomes the major player. This is just a blow-up of what we think happens when you get an infection. This is the epithelium, and this is the trauma. This is a break in the skin. The virus, as I said, cannot enter these cells. It has to get into these lower basal cells. And it does so through a series of interactions on the basement membrane, where ultimately the viral capsid will gain entry into the cell, into the early endosome. There's a process of acidification where, to the late endosome, where you begin to see the capsid breaking apart. And then the L2 DNA complex can escape from these late endosomes in the same time the L1 gets degraded in the lysosomal fraction. And the L2 DNA ultimately makes it into the nucleus, where you can initiate viral gene expression. Now, I've drawn this as a very sort of simple route by which the virus can get into the cell, but it's actually very, very complicated. There are many, from this late endosome, there are many different pathways that a cargo can be transported. And what we've been very interested in is trying to understand what is it about the virus that determines it takes this particular route. And I'm going to tell you about one of those aspects which concerns the choice of how fate determination at this step when the virus can escape from these late endosomes. And the way we went about doing this, or the way Martina went about doing this when she was in the lab, was to do a proteomic screen for L2 interacting partners. And what that means, basically, just simply, is, we take the L2 protein and we just ask, what cellular proteins can it interact with? And the striking candidate protein that we found was something called sortin nexin 17, or SNX17. So why was that particularly interesting? Well, sortin nexin 17 is a cellular protein, and its major function is determining the fate of proteins that go through endosomal transport pathways. So this seemed like a wonderful way in which the virus might hijack a known pathway, a protein that was involved in a pathway of endosomal sorting, so that it could use it for its own purpose of getting to the right part within the cell. Now, this protein, any of its cargoes, the proteins that it carries around the cell, if it cannot interact with them, they all get degraded in that lysosomal component. So they just cannot survive in the cell. And the other thing that was intriguing was that all the proteins that interact with this sortin nexin have this very highly defined amino acid sequence. And when you look at the HPV16L2 protein, that sequence is perfectly conserved in that protein, so it looks like a really good candidate. And if we make mutations in this, this is just a straightforward assay for protein-protein interaction. You can see the wild-type protein binds very strongly to this sortin nexin. The mutant that has destroyed this motif does not bind at all. So this looks like a perfect interaction with this cargo sorting machinery. So how do we go about saying, okay, we know these two proteins interact. How do we go about asking, is this important for virus infection? And what we do is we use so-called pseudovariant technology. And the way that works is as follows. You can take a plasmid, which will express the two capsid proteins, L1 and L2, and you can co-transfect that with that a reporter construct. In this case, something for luciferase, but you can use GFP. And you transfect these two plasmids into 293 cells. And what is absolutely amazing about this virus, just the very high level expression of these two proteins in the cells, they will spontaneously form virus capsids. And when they do that, they will also encapsulate this reporter DNA. So it's a really, I'm always amazed when people show me the results from this sort of thing, that you can just take two proteins, you put them in a cell, you put in a reporter construct, and you can generate these virus particles carrying these reporter genes. You can purify them quite easily, and ultimately you finish up with essentially papillomaviruses carrying a reporter construct. And this is what they look like. They look just like papillomaviruses that you would identify in a normal lesion from a person. And so you can use these reporters, these viruses, to infect cells, and you can measure reporter activity, both in a wild-type condition and also in a condition where you've destroyed expression of this sortin nexin 17. And you can see very clearly there is a dramatic reduction in virus infection. So you need this sortin nexin for infection to occur. The other way in which you can do the same experiment is to use a virus where you've mutated the L2 so that it cannot interact with the sortin nexin. And again, infection rates are dramatically reduced. So what is happening? Why doesn't the virus succeed in the infection when you cannot have this interaction? Well, the bottom line is, it's just like all the normal cargoes of this sortin nexin, the viral protein, the virus capsids, they just all get degraded in the lysosome. So the virus just cannot make it into the nucleus, it can't get out of the endosome, and all the virus gets degraded. So this association, the ability of the L2 protein to interact with this sorting Nexin 17, plays a critical role in the virus escaping from this pathway into the lysosome, but into this pathway where it can ultimately gain access to the nucleus. And really, this is just to summarize this first part. It's essentially just to sort of put it in words what I've just described. So you have a very unique interaction between the viral, this minor capsid protein of the virus, with a cargo sorting machinery of the cell, which then enables virus infection to be completed. So what I'd like to do in the latter part of this presentation is to talk about some of the events that take place at this stage. So we've just considered what happens right at the very beginning of virus infection. Now what we want to do is consider what is happening in stages where we ultimately get the development of a malignancy, and how are these two viral gene products contributing to the development of these tumors. Now, work from many laboratories over many years has really come up with this model of how these two viral oncoproteins contribute towards disease development. We know that in the very early stages, E7 plays a major role in driving proliferation of the infected cells. So you get an amplification of the population of cells that are infected with the virus. Now, under normal circumstances, when you get this unscheduled proliferation, the cell mounts responses to try and stop this growth. One of the proteins involved is a protein called P53. And E6 is very good at blocking this. So what this means is the two proteins contribute towards the development of immortalization, meaning the cells become immortal. They can just continue to grow and proliferate. What's also very clear, however, for tumors to actually develop requires additional changes. And one of the aspects of E6 function that I'd like to discuss today are the effects that it can drive within a cell, are changes such as effects on cell polarity. That's how a cell knows its top from its bottom. And cell-cell contact, so how well cells can stick together. And E6 is clearly a major player in these later stages of disease development, where you get ultimately progression to metastasis. And it's metastasis which is the fatal aspect of the infections with these viruses. So what I'd like to consider are what we know and what we think how E6 is contributing towards these stages of disease development. Now, E6, it's just to emphasize that it's a very multifunctional protein. It can interact with a huge number of cellular substrates. And not surprisingly, it has a wide variety of different biological activities. Now, when people talk about viral oncoproteins, they always tend to say, oh, it causes cancer, it does this, it causes cell immortalization. But the first question you always have to bear in mind is, what does it do for the virus? Because this is why these viral proteins have always evolved. It's to aid the virus to replicate. And clearly, some of these interactions of E6 are involved in genome maintenance and genome amplification. They're promoting the viral life cycle, which is exactly what you'd expect them to do. At the same time, they can also be subverted by the virus or by the E6 protein to bring about changes that can ultimately give rise to malignancy, such as immortalization and cancer induction in model systems. I mentioned at the beginning that there's a lot of different papillomaviruses, I don't want you to look at the numbers or anything on this, it's completely irrelevant. It's just to emphasize there's actually close to 200 papillomaviruses, but only about 10 or 12 cause cancer. So that leads to this stratification. We have so-called high risk, which are cancer-causing, which are like HPV-16 or HPV-18. And we have low risk. These only cause benign lesions, and they would be, perfect examples would be things like HPV-6 or HPV-11. So one of the questions that my lab and a lot of the field has been interested in is why do you have these papillomaviruses where some can cause cancer and some only cause benign disease? Obviously, it's got to be something related to E6 and E7 because these are the two proteins that drive the tumor. And we all spent many, many years looking for the different cellular substrates of these two viral proteins with the idea of saying, well, can we find something unique that might explain their cancer-causing ability? Unfortunately, in the vast majority of cases, we couldn't. And that's not surprising when you consider how similar the life cycles of these two sets of viruses are. What we did find, however, was that for the high-risk E6 proteins, those proteins derived from cancer-causing types, they had a unique capacity to target a set of cellular proteins that harbored so-called PDZ domains. So what is the rationale behind that statement? What I'm showing you here, these are different E6 proteins, all derived from viruses that cause cancer. And if you look at the extreme C-terminus, so this is the last 4 amino acids right at the extreme C-terminus of E6, you can see there's a very highly conserved stretch of residues. And this corresponds to a so-called PDZ binding domain. If you look in the low-risk papillomaviruses, those that don't cause cancer, you don't have this sequence present. So this is a molecular signature of oncogenic potential within these cancer-causing viruses. So what does this region of E6 do? What you can do, you can take your virus and make mutations here. So the only thing you destroy is that sequence. And then you ask, how does the virus behave? Well, it needs this region to be able to cause cancer. If you mutate this region of E6, it doesn't cause cancer in transgenic animals. If you make mutations in that region, it's also defective in its ability to induce transformation in model systems in the laboratory. It cannot transform cells. And, most gratifying, it's also important for the virus life cycle. If you mutate this region of E6, the virus cannot replicate. It actually loses its episomes and you clear the virus very effectively. So it clearly has a major role to play in all these different biological activities of the E6 oncoprotein. And what I'm going to talk about are 2 cellular substrates that we know are key players that are targeted by this region of E6. And why do I pick these 2? One protein is called disc large, the other protein is called scribble. Now what these 2 cellular proteins do is they actually regulate cell contact. So they regulate how cells stick to one another. They regulate cell attachment, so how a cell sits on a basement membrane, they control that. And they also regulate orientation or polarity, so how a cell knows its bottom from its top. And they provide this link between, this so-called link between polarity and proliferation control. I'll say a little bit more of that in a minute. But it's also important to note that a key feature of cancer progression and metastasis is a loss of loss of control of all of these aspects, with loss of contact inhibition, loss of cell attachment, and a loss of cell polarity control. So the working hypothesis being that E6, through targeting these proteins, is driving this progression to malignancy. This is cell polarity control. It's controlled. These are two cells. We have regions where you have cells, sites of cell-cell contact. And the cell polarity control module is controlled by three groups of protein. The SCRIB complex defines this region of the cell. The PAR complex describes the subapical. And then the Crum's complex defines the apical. Now, the only thing you need to know about this is that polarity control is determined by a series of interactions between these different components. And the key feature being the correct cellular localization and the correct level of expression. If you perturb the expression of any of these components, the functioning of the whole breaks down. And this in turn links to a whole variety of other pathways that essentially control cell proliferation. And we know that E6 can impact upon this pathway, particularly by targeting this group of proteins here. So what does E6 do to these proteins? I told you that the key point was their levels of expression, that they have to be expressed at a certain correct amount. Well, E6 actually degrades it very, very well. It just targets the protein for degradation. And you see that this occurs with HPV-18E6, to a somewhat lesser extent with 16E6, but not at all with 11E6, which cannot interact with these proteins. So it cannot affect their levels of expression. And this is reflected in the ability of these two E6 proteins to interact. The strength of association is a direct reflection of their ability to target this protein for degradation, with 18E6 binding very strongly, 16E6 binding a little bit more weakly. And we were sort of quite intrigued by that, because, you know, they have a very similar sequence, so you would think that these two E6 proteins would function in a very similar manner, would interact with these proteins in a very similar way. But in actual fact, what you can see is there is actually one slight difference. HPV-18E6 has a C-terminal valine. HPV-16E6 has a C-terminal leucine. And we asked the really naive question, could a single amino acid difference affect the function of these, this region of the E6 protein? So what we did was just do a very simple experiment where we just swapped the last amino acid. So we made HPV-16 look like HPV-18 and vice versa. And when we do that, the results are quite striking. So again, if we look at this dislodged protein, we're looking at its ability to be degraded. You can see that 16E6 does reduce its levels of expression, but 18E6 always does it better. If you make 16E6 look like 18E6, it becomes a lot more active. And the converse is true. If we make 18E6 look like 16, it becomes weaker. So just a single amino acid difference in the E6 protein in the C-terminal residue can have quite profound implications about the choice of substrate, about which cellular protein it's going to preferentially interact with. And so what this drove us to do was to do some crystallization studies in collaboration with Xiaojing Chen in China, in Southern California, to try and determine what was the molecular basis for E6 recognizing these cellular substrates. We obtained 3 crystals, one with another substrate protein, Magi1, one with DLG through this PDZ domain 2, which we knew was bound strongly in vivo, and another domain which we know that E6 does not normally recognize in vivo. It's a purely artificial construct. And when you look at that crystal structure, it told us a lot of information about how E6 can interact with these different substrates. And the bottom line is that it told us that within that C-terminal stretch of residues within the E6 protein, it isn't just the ETQV, the last 4 amino acids that are critical. It's actually a much more longer stretch of the protein, which tells us that substrate selection by these different papillomaviruses is really quite complex, and it tells us that different papillomaviruses, different viruses that cause cancer, will probably do it through targeting the same pathway, but possibly different cellular substrates. Can we do anything useful with this information? Well, one of the things that we can do is to take these structures and do in silico screening to look for compounds that we can use to try and block the interaction. And this is work that's currently ongoing. We have a number of compounds now that will block the association between E6 and these substrates. And we're trying to go along the line of seeing if we can develop these as fantastic research tools, but also tools that may be of potential therapeutic value. The other thing you can do with this sort of information is to actually go to your target protein, the protein you know that is being bound by E6, and look at what determinants in this structure are critical for E6 recognition. What this allowed us to do, in fact, was to define a residue within this region here that was critical for E6 to interact and target it. And we could make a mutation here such that it's resistant to E6, so E6 can no longer see it. And when we do that, you can see here the wild-type protein is very efficiently degraded by the E6 oncoprotein. The mutant with this point mutation is resistant. So we've developed a protein which cannot be targeted by E6. So what happens if we take that and put it back into a tumor cell, a cell which is derived from a cervix, which is cancerous? We know E6 is there, it's targeting these proteins. What happens if we put back a protein that is then resistant? And that's what, I don't think you can see this very well, but this is in HeLa cells, and we look at colony formation. With the wild-type protein, you still get proliferation because E6 can degrade it. When you put back this mutant, you can see you don't get any proliferation. So by putting back into the tumor cells the protein that is resistant to the E6, you can actually block the proliferation of those cells. So how does it do that? And this is an assay just looking at the levels of apoptosis in those cells. And the bottom line is that when you re-express this mutant that cannot be targeted by the E6 oncoprotein, you're inducing massive levels of apoptosis within these tumor cells. So this shows really, I think, very nicely that you can, and this just shows the quantification, that you really induce much more apoptosis in those cells. So this shows, I think, very nicely that at least with one of these substrates of E6, by using this really basic information on looking at the crystal structure, you've been able to generate a protein which is resistant to E6 targeting and use that to go back into the cells and say, what happens if I rescue this protein? And clearly, in this case, you actually induce apoptosis or cell death in the tumor cell, suggesting that this avenue of approach for trying to treat these tumors is actually very relevant. And really, this is really just to summarize what I've just said. PDZ domain targeting is specific for the cancer-causing mucosal types, and it does indeed represent a molecular signature for oncogenic potential. There's this pathway of polarity and proliferation control is targeted by many of the different cancer-causing papillomavirus types, but the details of it are somewhat slightly different. And that's really borne out by the structural studies that we've done. But it's also very exciting that we can now use this information as a way to try and develop novel therapeutics. And certainly in the case of at least one of these targets, restoration of expression results in cell death of the tumor cells. I'd just like to acknowledge the members of the lab that have been involved in different aspects of the work, and two really great collaborations. Xiaojing Chen, who I mentioned, with all the structural work, and a fantastic ongoing collaboration with Martina Brigant, who was responsible for all the life cycle infectious stuff that I told you about at the start of the talk. And of course, our funding bodies, without which we wouldn't be able to do any of this. And finally, I'd just like to invite you all to the DNA tumor virus meeting that we're hosting in Trieste this coming summer. So thank you very much again for the invitation, and thank you for...
So, thank you very much for the presentation. I think that this is a topic that affects all of us, so I'm sure there's many questions out there, so feel free to ask.
Elsa. Hi. Hi.
Very inspiring, and congratulations for many new results you showed. So I have actually, starting from just your last data about the therapeutic option of this protein, I was just thinking that how could be delivered that protein in terms of therapeutic option, and if maybe pseudovirus that are used for cancer vaccination could be used also in the therapy as carriers.
I think with the protein that the work I was describing about the protein, I think that's really more of a research tool. It's more to verify for our own peace of mind that if we can block these interactions or rescue the expression of the protein, we actually do get a phenotype which is potentially therapeutic. To address it from a purely therapeutic, I think you have to go down the drug through the drug screen that we're developing, and go down that route to try and get compounds that you could actually use to treat the tumors. I think delivery of the protein in whatever vector you decided to try could be I think it will be very tricky actually, because you've also got the added complication that if it got into the wrong cells, you cause as much trouble as you're solving. So yeah.
But actually, I have also another question about cell polarity.
Yeah.
If if you expect that during aging, such tissue could have impairment of cell polarity and therefore maybe more
I don't know.
to have more risk of infection or more prone to infection?
I don't know. I don't know if anybody's looked at polarity regulation with aging. I really have no idea. Could be quite interesting.
Then maybe I can ask one of more general questions. You mentioned that there is a huge amount of different types, which is unusual in a way, but there are also some oncogenic and non-oncogenic. And probably most of us will be in contact with oncogenic types during our lifetime, but still we won't get cancer at the end, not all of us. So what is the main trigger? Do we know?
I mean, nobody knows. I mean, nobody knows why some people develop cancer and some don't. It's clearly related to immune surveillance, so people are looking at the immune system. There's been a huge amount of work to look at HLA markers and MHC, and really none of that has given up anything really concrete. And you know, if you were to highlight one single biggest contribution that you could probably make to the field, is that because you get so many people infected, you know, you can get as many as 10-20% infected with a high-risk papillomavirus type, but less than 1% will develop a cancer. Now, how do you know, at the moment, all you have to do is you have to follow everybody and you have to keep going for tests to make sure nothing bad is happening. What you really need, though, is a test that could say, okay, of these 20%, this 0.1% are the ones that are really at risk, and the others we can forget about, because all they will have is a normal papillomavirus infection, and it will go away, and that'll be it. But at the moment, we don't have that.
But what do you think? What is the gain for these oncogenic viruses?
Sorry?
What is the gain? Why do they?
Don't think there's any gain. There's absolutely no gain at all. The point is that between these different virus types, one of the things I was trying to bring across was that although we stratify them into high risk and low risk, even within those, there's quite significant variation. But when you look at the life cycles of how a high-risk virus replicates its genome and a low-risk virus replicates its genome, you begin to see some clues as to why you have this difference. So a low-risk virus replicates its genome mostly in the lower part of the epithelium, in the cells that are already in the cell cycle. Okay? So it doesn't need to do the job of pushing, or it doesn't need to be as vigorous in driving cell proliferation. The high-risk virus types, on the other hand, tend to be replicating their genomes at a slightly higher level in the epithelium. So it needs to be somewhat more aggressive in how it targets cell cycle regulation. And I think it's for those, it's just how the viruses have evolved. They both occupy very successful niches. It's just that with the high-risk virus types, there is this potential to cause cancer, which is not something it wants to do. It's actually making it less fit, because once you do start to go down that process, the virus loses the capacity to divide and to replicate.
Do you have any other question? There is one there.
Actually, I just want your comment. Like, for example, some of the viruses, when they came, they're causing so many deaths. Suddenly they just appear like this Ebola, and they just suddenly cause so many deaths. Is there any research going on to avoid such kind of circumstances in the future, or?
Sorry, I missed the last question, the last part of the question.
Is there any research going on, like to avoid such kind of circumstances, like the which created this Ebola virus in African countries? Or?
I mean, obviously there's extensive surveillance going on to try and monitor virus outbreaks, and certainly within papillomavirus, there's an extensive monitoring network to look at different papillomavirus types and looking for potentially new viruses that might be problematic for humans. One of the interesting features linked to what you say, because we have the vaccine, we are going to see over the next 10 years or so HPV 16 and HPV 18 going down, because the population will be vaccinated and the virus won't be able to maintain itself as well. So you'll actually have fewer people infected. So one of the big questions is that if you clear those two viruses, are you going to leave a niche for non-vaccine papillomavirus types to become more prevalent? And so clearly, related to that question, there is a lot of work being done to follow very closely people who are vaccinated to see whether or not there is indeed other viruses coming in to replace the place of 16 and 18. Yeah.
Related to that question, why there are so many different HPV viruses? Just different tropism? It's not very common that you have 200.
They're extraordinarily successful. That's one of the reasons. And also, you know, if you they go back way, way back evolutionary. You know, dinosaurs probably had them. You know, we certainly, birds have them. And if we look, you know, and if anyone looks hard enough, you could probably find as many bird papillomaviruses as there are human papillomaviruses. So dolphins, you know, you name it, they're everywhere. And so they've essentially evolved with us, and they've changed over time, and new types have branched off, and so on and so forth. But 99.9 times out of 100, we don't even know we're infected. You know, we're all shedding papillomavirus. So, you know, and you don't know it. And so, in a way, they're perfect. They're perfect viruses because they just hang around and they don't cause any problem. We don't even know they're there. And so it's a given, you know, the immune system doesn't deal very strongly with them. So they've expanded in number hugely.
Okay. If there is no other question, I have still one very general in terms of tumor DNA, tumor viruses, tumor-causing viruses.
Yeah.
It's getting actually more and more known that more and more tumors are related to some viruses. What do you think in the future we'll know more about them? Even HPV, that's quite recently renowned as...
So you're saying that you think there may be other tumors where viruses may become...
Yes.
Yeah, sure. There's a lot of work being done recently, you know, using next-gen sequencing and deep sequencing and going in and the cancer, you know, the cancer samples are available for all the RNA-seq. So there's a lot of people looking very hard for new viruses that might be associated with cancers. And there are some candidates out there, but I don't see it growing massively. I think we're getting close. I think we're getting close to the end in terms of the viruses that cause cancer. Because, you see, you've had a fantastic experiment with HIV. So anybody who's, you know, HIV immune suppressed, all that group of viruses that I showed you at the beginning that cause cancer in people, in HIV-infected individuals, all of those cancers are a lot more prevalent. Okay? Whereas a lot of other cancers are not. So there's actually already the information there. You look in where you have HIV-infected individuals, and you see a big increase in a particular form of cancer, then you've got there's a smoking gun there about the possibility of a viral involvement or an infectious agent, let's say. And a lot of those they've been assigned.
Now I have a question.
I have one more general question about viruses.
Yeah.
It is known that for all viruses that some viral particles are infective and some are not.
Yeah.
And could you give I don't know, molecular explanation or why this is so, or a structural explanation? Or?
Well, I mean, I showed you how we produce these viruses in the lab, okay? And when, you know, you have to think that within a cell, you're producing millions of these things. There's thousands upon thousands of them. And basically, not every time does the in the case of papilloma, but you could apply it to any virus, not always do the components that comprise the virus come together in the right way. You sometimes get an empty capsid. You know, sometimes the virus will form itself and produce a coat, but there's no DNA inside it. Sometimes it might package the wrong DNA. You know, some of these viruses are very promiscuous. They'll package anything. I mean, papillomavirus will package anything that's around 8 kb will get packaged, whether it's DNA, RNA, you name it. So these are all the things that can give rise to defective particles. It's just that they tend to be rare. They're not as, obviously, for the virus, it wants to be as efficient as possible. So it makes a huge amount of coat protein, it makes a huge amount of DNA. And the chances are that in, 90% of the time you're going to produce things that are perfectly okay. But there'll be another 10% which are not. And it's a numbers game. It doesn't really care about that 10%. There's still enough that's going to give rise to a good infection. So there's no common mechanism of how a defective virus is generated. It varies from virus type to virus type. Does that sort of answer it? Yeah.
Okay. If there is no more question, then thank you very much again.
Thank you. Thank you very much.